Bismillah Rahman Rahim. Welcome back, guys. Welcome to another video lecture by Kami Biology. Today, in this video lecture, we are going to talk about genetic library or genomic library. Now, this is another video lecture which is also regarding to recombinant DNA technology. Now, if you guys remember in the previous video lecture in recombinant DNA technology, we talked the cloning techniques, we talked the overview of recombinant DNA technology, and we talked the vectors, and we also talked the screening and selectable markers. We all talked these things in recombinant DNA technology in my previous video lecture. If you missed that video lecture, it is present into my channel, you can watch it. Okay, so before going to explain genetic library or genomic library, you guys need to understand the overview, the vectors, the screening, the selectable markers, which is regard to recombinant DNA technology. Then the genomic library will be very easy for you. Okay, so let me raise this one portion and let me write. Right, genetic library. Now, what are the genetic libraries? As the name indicate, libraries. All of you better know about the libraries. Inside the libraries are those specific area where the different kinds of books are present. Okay, so if we apply this term on the genetic level, so it means that it is that specific area where our gene of interest, a lot of genes are present. Okay, so that area will be plates. That area you have plates is, let's suppose this is the library. Why? Because Petri plate I'm talking about. The Petri plate in this case is act as a library, and in this Petri plates we have different colonies which carry different kinds of genes. Okay, so we call that genes library. Okay, and now there are usually two types of the genetic libraries. One we call that genomic DNA library. Let me write genomic, and the other we call that cDNA library. Okay, now in this video we are going to talk about the genomic library. First of all, we are going to talk about we need to try and understand about the genomic DNA library. Then in the next video we are going to talk about the cDNA library. Okay, now as I told you that at genetic level the library are those where we can store our genes inside a bacterial cell on a Petri plates. Okay, now whenever we talked about genomic DNA library, the same things are here. So in this case what we are going to do, we just means we cut our gene of interest on a huge genome, and we loaded our gene of interest into a vector, and after that we deliver this vector to the host cell, and after some time we allow the host cell to grow on a Petri plate, and then we get a lot of bacterial cell. Now that's called as genomic DNA library. Why? Because each host cell can carry the recombinant DNA, and each recombinant DNA contain what? Each recombinant DNA contain our gene of interest, which is insulin genes or human insulin genes, or we can say chaperones, enzymes, different kinds of enzyme it may be contained. Okay, now we call that genomic DNA library. Now genomic DNA library is very important for our research for the molecular level, or in recombinant DNA technology it is very important steps. Now genomic DNA library, whenever we want to get genomic DNA library, so it contains two steps. Let me write. In the first steps we should prepare our genes. Preparation of genes. This is the first steps, and in the second steps we are going to do screening. Now by following these two steps we can get the genomic DNA library. But each steps are too much difficult from each other. Preparation of the gene is also difficult, and screening is also difficult. Screening is much more difficult than preparation of the genes. Okay, now how we can get the genomic library by following these two steps. First of all, we need to select the host cell, or no, not the host cell. We need to select the cells which contain our gene of interest. Let's say we select the human cells, bacterial cell, or we select plant cell. Those cells which we are interested, which we know that each cell contain our gene of interest. Let's suppose each cell contain the insulin genes. Okay, so we want to select each of the cell. Now once we select the cell, then what we are going to do, we should treat each cell with the detergent. Now once we treat the cell with the detergent, so then we get our gene of interest. So the genes is too much huge. So if I draw the genes, so it is large genes we have. So let's say this is our gene of interest. So a large genomes we get. Okay, that could be the bacteria genes, human genes, plant genes, okay, whatever. Okay, now once we get the genes of interest, then we treat these genes with restriction enzymes to create multiples, or we can say to create multiple genes. Okay, now we treat these genes with restriction enzymes. Now once we treat the genes with restriction enzymes, or we can say that we can, this is the first steps of the genomic DNA library. In the first steps, we are going to do the preparation of the genes. Now this is how we can prepare the genes. So we can say that we can prepare the genes by using recombinant DNA technology, or we can say we can prepare the genomic, means the first steps of the genomic library, or we can say the preparation of genes by using cloning techniques. Now once we apply the restriction enzyme on the genomes, then they convert the genes into small fragments. Okay, so we get our different pieces of our gene of interest. Okay, so we not only apply one restriction enzyme, we can apply EcoR1, BAMH1, Handy 3, these kinds of restriction enzyme we can apply on huge genomes. So there is not only one huge genome, a lot of genomes we have, a lot of cells we can treat it, due to which we get a lot of genomes. Now after some times what we get after some time, we get different kinds of genes. Let's say these are the gene, or these are the genes, pieces of the genes we get with this restriction enzyme. Now once we get the pieces or the fragments of the genes having the compatible ends, then we take vectors. Okay, now vectors are commercially available. Now once we take, then after that we take the vectors. All of you better know that there are different types of the vector we have: phasmid, cosmid, yak, bac, plasmid. But let's say in this case again we take the plasmid. Okay, usually plasmid are very important, or we can in molecular level, whenever we are doing recombinant DNA technology, we usually take the plasmid. Or okay, although we can also take the other vector as well. Now then what we are going to do, we allow this vector to receive our target genes. These are the target genes, and these are the human or genomes. Let's say genomes. Now after some times we allow the target genes and vector to combine with each other. We also treat the vector with the same restriction enzymes which we treat our gene of interest. Now then we add the ligase enzyme here also. Okay, now once we add the ligase enzyme and we add our target genes, then what actually happen, our target genes are now bind. So our target genes are bind with the vector, and then ligation happen by the ligase enzyme. Then all of you better know, in the previous video lecture we talked that vector are the delivery system. These are the vehicles that can carry our gene of interest. Now this is called as recombinant vector, recombinant DNA vector. Why? Because they can carry our gene of interest, or we can recombine our genes. Then the next step is that we should transfer this vector to a host cell. Okay, so this is, let's say, the host cell. The host cell could be a bacterial cell, it could be a yeast. Okay, so E. coli, we can say E. coli, or we can say yeast, any kind of cells which can easily take the vector, the recombinant DNA vector. Now once the bacterial cell receive the vectors, so let's say some of the bacterial cell can receive the vectors. Okay, now once the bacterial cell receive or host cell receive the vector, then our next step is that we allow the bacterial cell to grow on agar plate. Then we allow each of the bacterial cell onto the agar plates. Let's say this is the agar plates. Then each of the bacterial cell can grow in the form of each bacteria can multiply and dividing, and we get a lot of bacterial cell in a form of colony. So we get a lot of colonies here. Okay, so this is how we can prepare our genes, our target genes. This is how. Okay, as I told you that it is very difficult step. Now how it is difficult steps? You know, the separation of the separation of the genome is very difficult. Whenever we treat the human cell, or whenever we treat any kind of the cell with detergent, so it not only rupture the membrane, it can also rupture your genome as well, or we can say it can also degrade your genome as well. So it is very difficult. Next problem is that when you get the target genes, then some of the target genes can make a complementary with means there are the complementary end or complementary sequences of the target genes with each other. So the target gene will bind with each other, some of them, if the sequences are complementary to each other. So each target genes will bind with each other. This is another problem whenever you are preparing the genes. Sometime what actually happen, the vector which we treat with the restriction enzyme, some of the vector can get our gene of interest and some of them are not get. Okay, it doesn't means that all the vector will get the gene of the interest. Now there are some vector that can take your gene of interest. And next thing, you loaded your then you deliver each of the recombinant DNA vector to the host cell. Okay, so some of the host cell can receive receive your recombinant DNA vector. Not all the cells receive. Okay, that's also a difficult step. So you are doing these steps again and again to make sure that the bacterial cell can get the recombinant DNA vector. You are doing these steps so many times to make sure. Okay, now this is very difficult. That's how we can say that it is much more difficult. But let's say you doing these steps many times. After some times, then you make sure that your bacterial cell or your recombinant DNA is now inserted into a bacterial cell, and finally you prepare the genes. Once you complete this step, then the next step is the screening. It is much more difficult than preparation of the gene. It's more difficult than preparation of the gene, we can say like that. Now how it is difficult? Why? Because it is this much difficult because bacterial cell is too much small. You can't see it with your naked eye. Genome is also smaller than bacteria. Then vector is also smaller, as well as your target gene is too much smaller. You can't see that what actually happening. Whether your gene of interest is cut, whether the vector is cut off, whether the vector is receiving the same things, whether the bacterial cell receiving the whole of the things, you don't know about that. So whenever you need to understand that which cells receive your target genes, your target genome, which cells. For that purpose you are doing the screening. But screening is difficult. How? Because whenever you are using the screening method, so it create a lot of problems. Let's say for the screening we use fluorescence techniques. Sometimes we use, there are different types of the screenings. Inshallah, we will be talking in some other videos lecture on the computer screen. We are going to talk about this different types of the screening. Okay, so let's say here I will just explaining two types of this, explaining just two example of the screening, not in a detail, just a little bit. We have a techniques of fluorescence technique, fluorescence techniques. Try to remove the spelling mistake. Techniques. Okay, now we are using two fluorescence techniques. In this case, what we are actually going to do, we use a single probe, or we can say we use probe. Each probe, or we are using a single strain of DNA. In this case, we use single strain of DNA which contain the radioactive substance, or which contain the fluorescent substance. Then we add each fluorescent substance, or each probes. This is actually a probes. Then we add the probe with the in each colony, but we not directly put the probes. Why? Because at that case the DNA is double stranded. So to make the DNA single strand, we use some kinds of enzyme. We put some enzymes here. We put some enzymes here. Now once we put the enzymes, so the DNA, let's say in this case, the DNA which are present in each colony cells, let's say, they become single stranded. So the DNA are now become single stranded. And after some time, then we loaded the probes here. Now once we loaded our probes here, so what actually happened, our probes are now they are complementary to this our target genes. They are complementary to our target genes. Now whenever they are complementary to our target genes, so we get a DNA RNA hybrid. We get the hybrid. Okay, now this is called now this technique is called as a colony hybridization assay. Now in this technique, let me once again tell you, once the probe and our target genes are complementary to each other, they are matching with each other, so then a fluorescence or a signal is detected, or we can check the signals, we can check the fluorescences through which we can know, through which we can ensure that each colony or each bacterial cell can contain our gene of interest. But it is not too much simple. Why? Because sometimes what actually happen, the probes which we use in this case, not only single probes, we use a lot of probes here. So sometimes each probes are matching with each other. This is the problem. Each probes are matching with each other, due to which we can't determine that whether this probe are matching with our own DNA or no, whether it is matching with the probes, because it's maybe the possibility that the probes, different kinds of probes which we are using here, may be complementary to each other. That's the main problems whenever we are doing the screening steps in this case. Now, okay, sometime we are using another techniques to reduce the errors of to reduce the error during screening. We usually called as we use antibiotic resistance genes, sometimes also antibiotic resistance genes. Okay, now what actually happened in antibiotic resistance genes, we already explained each of the things in our previous video lecture in genetic or selectable marker. We already talked the antibiotic resistant genes. Now antibiotic resistant genes, we add our antibiotic resistant genes to the vector. Now once we add antibiotic resistant genes to the vector, then it create resistance genes which can against to the antibiotic, due to which the bacterial cell can survive. So once we get the colonies, but we don't know which colonies have exact our gene of interest, we should make another plate. For the identification of our gene of interest, we make another plate. let's say this red plate shows that it contains antibiotic. In this case, let's say they contain ampicillin antibiotic. Ampicillin antibiotic. So we loaded our our bacteria to that plate. Now once we loaded the bacteria at that plate, so then what actually happened? Those bacteria which contain our gene of interest will grow. Why? Because they contain the antibiotic resistant genes which we use as a selectable marker, which is actually the screening. Okay, now once we loaded the bacterial cell and once our bacterial cell grow having the antibiotic resistant genes, then we make sure that this is our this is the bacteria which contain our gene of interest. Okay, so we also use this antibiotic genes for the screening method. Okay, we can also doing this. There are some other screening method as well, but these are the main things. Inshallah, I will explain the screening, different techniques of the screening in some other video lecture. Now, once the bacterial cell are grow, okay, let's say the bacterial cell are grow here, but still this technique is also difficult. Why? You know, whenever we using the antibiotic resistance genes in this case, so there is a possibility that is the restriction enzyme which we use for the cleavage to cleave our target genes as well as to cleave the vector. Sometimes this restriction enzyme can also cleave the antibiotic resistance genes, because this is the possibility. The EcoR1 or HEND3 or BAMH1 restriction enzyme, let's say these restriction enzyme have the cleavage point. Maybe they have the cleavage point at the antibiotic resistant genes. This is the possibility maybe happen. So if they have if they recognize their cutting sequences at that antibiotic resistant genes, so antibiotic resistant genes will cleave out, and due to which the cells will not grow. This phenomena will also happen. Once we get, let's say, our every experiment is now going in a good way, and finally we get the bacterial cells or the host cells which can contain our gene of interest. After some time, what actually we are going to do, we store each bacterial cell. Now once we get the our bacteria or host cells having our gene of interest, then we make a glycerol stock to preserve each bacteria or to preserve each host cell or any kind of cell on the glycerol stock, or some other chemicals we can make it to preserve our bacterial cell or host cells, okay, for years or for months. Now once we make our genomic library, then what are the importance of the genomic, why we prepare, why we usually select the genomic library. Now for example, let's say what actually we have. Let me explain the importance of the genomic library with the help of example. Now what actually we have, let's say there are a bookshop, okay, a huge bookshop in the market, and you go to the market and you just want to select a specific book. You want to select means you want to select a book of chemistry, biology, physics, environmental science. Let's say these kinds of books you want to select, and then you store each of the book into your room, which contains bookshelf, and then you store each book into a bookshelf, okay, and then you save each book into means at that specific room. Now after some time, whenever you need, you can choose that book and you can study that book, okay, whenever you need. But for that purpose, you don't go to the shop again. Why? Because you already store each of the book. Just like that, when you already make the genomic DNA library, then you can select each of your gene of interest. You can separate your gene of interest and you can run it on gel electrophoresis or any electrophoresis technique. You can separate your genes and then you can make your proteins. Let's say in this case you can make the insulin proteins and you can use it for your experiment. Means you can use it for the disease like diabetes. You can use it for diabetes disease, okay. This is how we can use the genomic DNA library. You not go to the, or you means you not repeat the same steps again. Means you select the cell and you separate the genome, and then you using the same steps and doing again and again, and due to this way you can also face difficulties, also you can face more problems. So to reduce these problems, to reduce each task or each difficulties, what you are going to do, you just already make the genomic DNA library. You just select your gene of or you just select each of the bacteria which can contain your gene of interest, and you can proceed it on a gel electrophoresis, or you can separate your DNA or any kind of thing, and you can proceed it, and finally you get your product. Okay, that's how we can use genomic DNA library to prevent the time, or to prevent the consumption of our time, or they can use for the prevention of the time. Now sometime whenever we want to prepare, or whenever we want to get the genomic DNA library, we always use a huge vector. Okay, in this case we usually use a fetch vector back. Let me write for the genomic DNA library, we usually use a fetch vector cosmid bacterial artificial chromosome, or we can say BAC. We always use each vector. Why? Because each vector have the ability to carry multiple gene of interest. Okay, now when you use the plasmid, so there are the problems with plasmid, because they can only take a few amount of gene of interest. So that's in a sense about the genomic DNA libraries. I hope you get the idea about the genomic DNA libraries, and if still you have any kind of confusion or question, if there is something missing, you can write it into a comment. Inshallah, I will give you answer as soon as possible. So in the next video lecture, we are going to talk about cDNA library. Okay, to complete the lecture of genetic library. So I hope you understand. Thank you so much for watching.